Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic control and coordination. So in the previous lecture we were studying about the roles of the different hormones. So we studied about the functions of the auxins as well as the gibberellins, and in this lecture we'll be studying about the role of the cytokinin, ethylene as well as the abscisic acid. See, we have already seen some of the roles of the growth promoters, which are auxins and the gibberellins. And the third growth promoter is the cytokinin. So let's start with this lecture. So again, I will give you an idea about the phytohormones. You already know how we can define phytohormones. The phytohormones, they are also known as the plant hormones because phyto is related to the term plants. Now, these are the chemical substances synthesized by the plant that regulate various physiological processes, including growth, development, as well as response to the environmental stimuli. So, when we were talking about the auxins and the gibberellic acid, so what they do? They help in the promotion of the growth. How they help in the promotion of the growth? By inducing the cell division. Okay, by inducing the cell elongation. Okay, and we have also seen in the previous lecture that how they induce the cell elongation. So we can say gibberellins and the abscisic acid and the auxins, they induce cell division, they induce the cell elongation. And if we talk about the auxins, they induce root formation also, or root initiation, or we can say adventitious root initiation in stem cuttings. They are also involved in root differentiation. Okay, if we talk about the gibberellins, okay, gibberellins, they were involved in the stem elongation. And we also studied about the process of or the phenomena of bolting. Gibberellins are also involved in the cell elongation. Okay, and they also promote the seed germination. Okay, by breaking the seed dormancy. They promote the seed germination also. Clear? Now, let's talk about the cytokinins. Okay, so what are the functions of the cytokinins? So cytokinins, they also have a lot of important physiological effects, okay, on the plant growth. Now let's discuss about them. First, major importance of cytokinin is the cell division and the delay of the senescence. So these are the most important function of the cytokinin. So one of the main function of cytokinin is in the cell division, okay, more particularly in the cytokinesis. Okay, one is karyokinesis, means the division of the nucleus. Another one is the cytokinesis. So during cytokinesis, okay, or you can say cytokinins, they promote cytokinesis and they are also involved in the cell division. Because of cytokinesis, two cells will be formed. Okay, so karyokinesis is followed by cytokinesis, and cytokinesis is promoted by the hormone cytokinin. Clear? Now, next, they are also involved in the secondary growth. Okay, secondary growth means you can say lateral growth. Secondary growth means the lateral growth. Now, when we were discussing about the functions of the auxins, we saw that the auxins, they promote the apical dominance. And what was apical dominance? The inhibition of the lateral bud growth, okay, in the presence of the apical meristem. This is known as the apical dormancy, where the lateral growth is inhibited. And the lateral growth inhibited because of the presence of the auxins that are produced in the tips or the apical meristem, such as the shoot apical meristem. Now, if we want to promote the secondary growth or the lateral growth, what we can do, we can either remove the shoot tip. Okay, so if we remove the shoot tip, no oxygen will be present and the lateral growth will then start. And another method is to apply the cytokinins. So cytokinin application, okay, it helps to overcome the apical dominance, okay, and helps in the promotion of the growth of the lateral buds. Clear? Now this is also used in the this also have lot of commercial importance. Clear? Now next important role is the senescence. So cytokinins, they help to delay the senescence. Senescence, you can say, is a word very similar to the aging. Okay, after senescence, the living organism will die. So what happens? The organism will grow, it will differentiate, and then after differentiation, it will undergo maturity. So maturity occurs, and after maturity, the person will start aging, or it will start senescing, and after senescence, it will die. Clear? So this is the life pattern, or you can say the life cycle. So first, the maturity is achieved. Okay, after maturity, then senescence begin. Okay, after senescence, there is death. So these cytokinins, or the application of exogenous cytokinin also, okay, if we apply cytokinin externally, then it will delay this process of senescence. So it can help to increase the life, all right, of a, you can say, leaf, okay, or the plant organ. Okay, now you must have seen, if you apply this cytokinin on a particular region of the leaf. So suppose if this is a leaf, okay, if you apply this cytokinin on this region, in this region you have applied exogenous cytokinin. Okay, here you apply cytokinin exogenously, means externally you are applying. Then what will happen? This area will remain green. So this area, it will remain green. Why it will remain green? Because here the chlorophyll molecule will be retained for a longer period. That's why this area will remain green and the rest of the area will turn pale. Okay, rest of the area will turn pale, or it will turn yellow. Why? Because here there will be a degradation of the chlorophyll, and only this area on which the cytokinin has been applied, it will remain green. Why? Because the chlorophyll molecule, they will be retained here for the longer period. So this is the role of the cytokinin. Okay, so what will happen? All the food materials, okay, and all the nutrients will be concentrated in this region. Okay, you can say all the nutrients present in these leaves, okay, if these nutrients are present in the leaves, they will be concentrated in this region. Okay, so this region is nutrient rich, all right, so it can synthesize all the molecules such as chlorophyll, because all the material is available, and this region will remain green. So this is a very important role of cytokinin. It helps to delay the senescence or inhibit the senescence, means it delays the aging process. Clear? Now let's talk about the next function. Next is the morphogenesis. It also helps in the morphogenesis. So a natural balance between auxins and the cytokinin is responsible for differentiation of stem as well as the root. And this process is known as morphogenesis. There is a development of a certain structure, okay, or there is a change in the morphology, okay. Due to that, there is a change in the morphology. All right, this is clear. Now, there should be a balance between the auxin and cytokinin. So whenever we are doing tissue culture, so we add both auxin and the cytokinin. So auxins we have seen in the previous lecture. What was the function? Root initiation. Okay. And cytokinins, they are involved in the initiation of the shoot. So what happens? But the ratio of the auxins as well as the cytokinin, this decide whether the root will be initiated or shoot will be initiated. So the ratio of the root, ratio of the auxins, okay, ratio of auxin to cytokinin, this will decide whether the root will be initiated or shoot will be initiated. So if the level of auxin is high in the culture media, then what will happen? Root will be initiated. Root initiation will take place. But if the level of cytokinin or the concentration of cytokinin is more, okay, if cytokinin is more, then what will happen? Shoot initiation. Okay, so we can say for shoot initiation, cytokinin should be high and auxin should be low auxin. Okay, then it will promote shoot initiation. Whereas when auxin level is high and the cytokinin concentration will be less, we can say cytokinin is less as compared to auxin, then there will be root initiation in the callus. Clear? So when cytokinin are in excess, lateral buds will develop. So cytokinins, they help in the secondary growth also, and they also help in the shoot initiation also. Clear? So lateral buds, you can say shoot initiation also, and you can also write here lateral bud growth. Clear? While the roots are formed if the relative concentration of auxin is more. Clear? So this is known as the morphogenesis. So initially we have the callus. So what is callus? Callus is the undifferentiated mass of cells. It is the undifferentiated mass of cell or cluster of cell. Now what happens? We add hormones such as auxins as well as gibberellic acid. So we have added auxin in this media, and then we have added cytokinin also, and we have added other hormones also. Okay, so now the ratio of auxin to cytokinin will determine whether there will be root initiation or whether there will be shoot initiation. Clear? So this was the very important function of cytokinin. The next function is the induction of the flowering. So what happens? Cytokinins, they play a very crucial role in initiating both the direct shoot regeneration and flowering process. So we can say the cytokinins, they help in the induction of the flowering, okay, in plants such as Scoparia dulcis. So in Scoparia dulcis, it has been observed that the application of the cytokinin, it helps in the induction of the flowering as well as the shoot regeneration also. Clear? The next role is the callus growth. What happens? The exogenous application of the auxins and the cytokinin induces callus in the various plant species. So in the previous slide, we have studied, then when we add these hormones, auxins and cytokinins to the callus, then it will lead to the initiation of root or the shoot depending on the concentration of these two hormones. But what happens in some plant species, the exogenous application of auxin and the cytokinin, they induce the callus formation also. Okay, callus formation is the formation of undifferentiated mass of cells. Clear? So this is another role of the cytokinin. It helps in the induction of the callus. How they induce? Because they induce cell growth as well as cell division also. So that's how they induce the callus growth also. But this is observed in some plant species. Clear? Now let's study about the another hormone, ethylene. So ethylene, this is the gaseous hormone. Okay, this is the gaseous hormone, and it is what? It comes under the category of growth inhibitor. All right, it comes under the category of growth inhibitor. So ethylene, this is the gaseous plant hormone which is produced by almost all the fleshy fruits during ripening. So that's why ethylene is also known as the ripening hormone. Okay, because this is produced during the ripening, so it helps in the ripening process. Okay, by increasing the rate of the respiration, and that is known as the climacteric respiration. Clear? So you must have seen in bananas, it helps to ripening. It helps in the ripening of the banana as well as the other fruits. So that's why it is also known as the ripening hormone, and it is produced during the ripening. Now, and how it induces the ripening? By increasing the increasing what? Increasing the rate of respiration. That is known as the climacteric respiration. Okay, that is known as climacteric respiration. You will talk about this term in the higher classes. Now, ethylene is autocatalytic in nature, means self-catalytic. Means, okay, it can. So ethylene is autocatalytic in nature. Okay, means self-catalytic. Clear? Now, the higher concentration of oxygens, it induces the ethylene formation. So whenever there is high concentration of oxygen, then ethylene formation can occur. Clear? Now, let's study about the important functions of the ethylene. So these ethylene, they are known as the fruit ripening hormone. So first function is the fruit ripening. So it is used for the artificial ripening of the fleshy fruits in the shops. Okay, so if you want to ripen any fruit, okay, so if you want to ripen the unripe bananas, then you can spray it with the gaseous hormone ethylene, and then it will ripen in some days. Now next is the abscission layer formation. So what happens? Ethylene accelerates the abscission of the leaves, flowers, and the fruits. So when we were talking about the oxygens, we studied about this abscission. Abscission is what? Abscission is the shedding of the leaves or the detachment of the leaves, fruits. Okay, leaves as well as the fruits. And this shedding or the detachment of the leaves of the fruit, this takes place due to the development of the abscission layer. Now, when we were talking about the oxygens, it prevents the abscission in the young leaves as well as the buds and the fruits, but it promotes abscission in the mature leaves and the fruits. And similarly, ethylene also, it accelerates the abscission of the leaves, flowers as well as fruit, means it accelerates the detachment or the shedding of the leaves, fruits and the leaves, flowers and the fruit by the formation of the abscission layer. And I have already told you, then when the abscission layer is formed, then what happens? The nutrient supply is cut off to the leaves, fruit and the flowers and the fruits, and then these parts of the plant will fall. Now next, let's study about the role of ethylene in senescence. So ethylene, it promotes senescence. So this is a role antagonistic to cytokinin. Cytokinin, it prevented the senescence, but ethylene, it induces the yellowing of the leaves and the downward bending also of the leaves. Okay, downward bending can be due to the changes in the turgor pressure. Okay, this can be due to the changes in the turgor pressure of the leaves. This turgor pressure is developed due to the presence of the, you can say, water in the cells. Clear? So it is inducing yellowing, and yellowing is induced by the lack of the chlorophyll. Okay, this is also known as chlorosis, breakdown of the chlorophyll. Okay, what is known as chlorosis? So that's why ethylene is known as a growth inhibitor. Okay, that's why ethylene is known as the growth inhibitor. Mostly it is inhibiting the growth. Okay, and the breakdown of the chlorophyll, okay, this is known as chlorosis. Clear? Now, growth. What about the growth? It promotes the transverse expansion, but it inhibits the longitudinal growth. Okay, means it promotes the lateral expansion, but it inhibits the longitudinal growth. Clear? Next is the flowering. So it induces flowering in the pineapple. Okay, so in some plants such as pineapple, it induces the flowering. Now, what about dormancy? It breaks the dormancy of several organs of the plant except the lateral buds. Okay, so it helps to break the dormancy, okay, of several organs of the plants. So we have studied about the seed dormancy, rhizome dormancy, tuber dormancy. So ethylene, usually it will help to break the dormancy, means it will help to germinate, okay, or the growth of the several organs of the plant, but it doesn't help to break the dormancy of the lateral buds. Clear? Now, this ethylene is also used for the induction of the femaleness. Clear? So it is also known as the feminizing hormone, or it has the feminizing effect. So what happens? So application of the ethylene, it increases the number of the female flower, such as in cucurbits. So in the previous lecture, we have studied about the role of a hormone that induces the maleness. Okay, but here ethylene, it is increasing the number of the female flower, means it is inducing the femaleness. Clear? Now this was the end of the roles of the ethylene. Now let's study about the another growth inhibitor known as the abscisic acid. So abscisic acid, this is also known as the growth inhibitor. Okay, it is also known as the stress hormone. Okay, it is also known as the stress hormone because it is produced during the stress conditions. That's why it is known as the stress hormone, because it is produced during the stress condition. And what are the stress? Stress can be biotic stress. Okay. What are the biotic stress? Or you can say environmental stress also. Conditions. All right. What are the stress conditions? One can be drought. Okay. Then another can be salt. So these are all the abiotic conditions. Okay. Excess water also is a kind of stress. Then high temperature. Okay. So it helps the plant to survive also because they are produced under the stress. That's why it is known as the stress hormone, but usually it is a growth inhibitor. So Abscisic acid is also known as the stress hormone of the plant, and earlier it was known as the dormin. Okay, so if you talk about the discovery of the abscisic acid, it was discovered 3 times independently. Okay, and later it was given the name dormin because it induces the dormancy. So it has negative effect on the growth. Okay, and the name dormin was given initially. Now we know that this dormin, the new name is the abscisic acid. Clear? Now, Endicott and his co-workers, they isolated a substance from the young cotton balls and named it as abscisic acid. Clear? Now, this acid is now isolated from the dormant seeds, okay, buds and the other parts of the plant. Okay, because they induce dormancy. Okay, then what the researchers they observed, or what they concluded, that in the dormant seed, buds, as well as the other parts of the plant that are dormant, that are not growing, abscisic acid should be present there, and they were able to isolate this abscisic acid from this dormant seed, buds, and the other parts of the plant. So abscisic acid is known as the growth inhibitor. Okay, abscisic acid has no stimulating effect on any aspect of the growth, means it doesn't have any positive effect, okay, on plant growth. It has all the negative effect. Okay, clear? Now let's study about the functions of the abscisic acid. Now first let's see the role of the abscisic acid in the plant growth. Abscisic acid is also abbreviated as ABA. So ABA has the antagonistic property to growth promoting hormones. Antagonistic property means the opposite property. Okay, so the plant promoting hormones are auxins, gibberellins, and the cytokinins. So abscisic acid or the functions of the abscisic acid is opposite to all these plant growth promoting hormones. Okay, thus the abscisic acid, it keeps the growth under check because it has a negative effect on the plant growth. Now let's have a look at the role of abscisic acid in the dormancy. So as I've told you, it will induce dormancy. So abscisic acid, it induces dormancy in the bud towards the approach of unfavorable conditions. So whenever there are unfavorable conditions, okay, such as I've told you, drought, high temperature, okay, or salt conditions. So these are the kind of stress, okay, and these are unfavorable conditions. So the seed cannot germinate. So what happens? Abscisic acid is secreted during these unfavorable or the stress conditions, and this will induce dormancy in the buds. And whenever the favorable conditions, they will arise, okay, then the seed will start germinating, and then gibberellic acid will be synthesized because gibberellic acid helps in the breaking of the seed dormancy. And also here, if you want to break the seed dormancy of these seeds, okay, then you can apply exogenously gibberellic acid, and then it will lead to the germination of these dormant buds as well as the seeds. Clear? So abscisic acid causes natural dormancy in seeds as well as the tubers. Clear? Now abscission, let's see the role of abscisic acid in the abscission. So abscisic acid, it promotes abscission in the leaves, flowers as well as the fruit, and it causes aging in the plants. Okay, so it promotes the aging or the senescence. Okay, you can also write here, it promotes the senescence. And which was the hormone that prevented the senescence? It was cytokinin. So you can say abscisic acid here is playing the opposite role as that of the cytokinin. Alright, it is promoting the abscission, okay, by the formation of the abscission layer again, okay, and it also causes the aging of the plants. Clear? Now another role is in wilting. So what happens? During the drought, it closes the stomata and checks the lot of water by the transpiration. So you can say it prevents wilting. Okay. It prevents wilting. So as I have told you, abscisic acid is a stress hormone. So drought is a kind of a stress. And abscisic acid is produced during the stress condition. So what happens? During the drought, there is the lack of the water. So because of this, abscisic acid produced and it helps to close the stomata. Okay, if the stomata is closed, then there will be no loss of water through the process known as transpiration. Okay, so it checked the loss of the water by transpiration. Clear? And you know about the stomata. What are stomata? Stomata are the tiny openings that are present in the surface of the leaf, or you can say these are the tiny openings that are present in the epidermis of the leaves, and they help in the exchange of gases. Okay, one role is in the exchange of gases. And another role is what? The transpiration. Clear? Now, this ensures the survival of the plant. Okay, so why? If it prevents the wilting, then what will happen? The plant can die because of the loss of the water. So abscisic acid, it closes the stomata, which prevents the loss of the water through transpiration, and that ensures the survival of the plant. Clear? So next role is the resistance. So it promotes the cold hardiness. Okay, it helps to induce, or it promotes the resistance. It makes the plant more tolerant. Okay, so it induces the tolerant and it promotes the cold hardiness. Clear? So that these are all the survival strategies. So during the stress conditions, what happens? This abscisic acid is produced, and okay, we have seen one example. It prevents the wilting. Okay, and it also promote the tolerant in the plants, so which helps the plants to survive in the unfavorable conditions. And whenever the favorable conditions they return back, then the plant it start germinating. Okay, the rest of the hormones are produced. All right, and the plant will resume its germination as well as the growth and the development. Clear? Now, next role is in flowering and the seed development. So, abscisic acid, this is known to inhibit the process of the flowering, fruit formation, and the seed development. So, you can see here, these are all the negative roles. It is inhibiting. All right. So, it is inhibiting the flowering process. All right. Fruit formation is also inhibited. So, if the flower is not produced, then the fruit will also not be produced, and there will be no seed development. Clear? So you can say there will be no reproductive phase. There will be no reproductive phase if you apply the abscisic acid, because induction of flowering happens during the reproductive phase. Okay, so if the flowers are inhibited, so abscisic acid inhibit the flowering process. Okay, fruit formation will be inhibited and seed development will also be inhibited. Okay, but there are some techniques to induce the flowering also. So we have seen about the photoperiodism, where we have studied there was induction of the flowering depending on the relative length of day and night, and the other process was vernalization, where the plants were treated with cold, or the cold treatment was given to the plants, means the plants were kept at 4 degrees Celsius. Okay, and that helps to induce the flowering. Okay, so all these processes can be reversed. So you can inhibit also, okay, using these hormones, and you can promote the growth of the plant as well as the plant parts using the other hormones which are growth promoters such as auxins, gibberellins, as well as the cytokinins. Clear? So in this session, we have talked about the functions of the cytokinin, ethylene, as well as the abscisic acid. Clear? Now, if you talk about all the lectures till now, You should know about the how you should define coordination, then how the coordination takes place in the plants, and what are the movement or what are the types of movement in the plants. Then you should also know about the phytohormones. Okay, so what are phytohormones? You should know the definition of the phytohormones. You should know the discovery, how these phytohormones were discovered. Okay, then you should also know what are the roles of this, or what are the functions of these phytohormones, and what phytohormones come under the category of growth promoting hormones, and what hormones come under the category of the growth inhibitor hormones. All right, so I think you'll be able to answer all the questions that are related to these topics. Now in the next session, we'll be studying about the control and coordination in the higher organisms such as humans. Okay, so I hope you enjoyed this session. Thank you very much for listening. We'll meet in the next lecture.